This computer. Anyway, hello everyone and welcome. Today we're going to have a presentation by Deborah Neher, who is a soil ecologist and professor emerita at the University of Vermont. She gained formal education in plant pathology, plant population ecology, and environmental science. And throughout her career, Deb's research has focused on developing biological indicators for soil health. So if anybody is joining late, and if you have questions, you could put them in the chat. I will probably repeat that later. But yeah, why don't we get started?
All right. Thank
You
you
want
for
to
that
share?
great introduction. And the fact that I have education across the plant side and the pathogen side, and also the ecology with the beneficial, kind of gives me a little bit of a unique perspective on soil health. So I hope that comes across today, and also indicates kind of the breadth of questions you're welcome to ask at the end. So before I start to show my slides, I just wanted to mention that I could really relate to grain. I grew up on a wheat farm in Northwest Kansas. I was fourth generation. And when I was growing up, so it'd probably be like middle school or so, I started learning how to mill flour because I like to bake bread. And there's nothing like fresh flour in fresh bread. And baking bread is still one of my favorite baking hobbies and projects. So with that, I'm going to share the screen here. I assume that shows up. Okay. So the topic today is, focus on soil biology and how it's related to soil health. Wanted to start out with, general overall characteristics of healthy soil are really, focused on how do we promote healthy plant growth. That's the end goal. And so I think of it as kind of a three-legged stool. We need soil to be chemically balanced. So you're meeting the nutritional needs of the plants, but not to have excesses. And we also need soil to be porous, and in other words, not compacted, porous, and have a range of pore sizes. And we also need a living matrix of soil biology as well as high organic matter, because they are involved in the processes of nutrient cycling and organic mass. Well, organic matter itself provides not only nutrients, but also microbes. And soil biology is frequently overlooked in standard soil health assessments. It's a relatively young discipline compared to soil science. Soil science was really recognized as a discipline in 1860s, where soil biology as a discipline was not really recognized about till 1970. So about a century difference here. And the reason I mention that is because the fact is these have been overlooked in the soil health is because we've had lack of tools, because they're microscopic in size, and all the creatures you see on this slide here are all microscopic, and you really need to see them either under a light microscope or an electron microscope. So trying to change my slides here. Okay. So here's a couple photos of soil. The one on the right is actually soil from an old field, and the one on the left is more of a diagram. But what to show the different features. But what I want to point out here is thinking about soil as a habitat for life. For life, we know all organisms are going to need some air, or oxygen. They need moisture, and also a source of energy, which we would typically just think of as food. So when we think about it, soil is providing all of those. Healthy soil is. So soil is a porous fabric, and we can see kind of the gaps here. You'd think being dark that it would seem more like a solid, but in fact, it's got you want it to have a lot of openings, and these pores provide a balance of the air and water. These openings are the pores are the homes really of the soil biota. And the network of the paths really determine how well they can disperse through this matrix. So on the left, you can see there's a number of soil organisms here illustrated with the threads of hyphae and let's see. And we can also see there's water in here and air balance as well. And then there's some little, what we call it, some of the microscopic animals that are residing in there. On the right-hand side, the blue in this slide is really open space. This is the pores, and it's a resin that we put in so that we could take a physical slice of soil. And the tan elements there are actual mineral particles like sand and silt or clays. And the dark material is organic matter, or there's in the lower left-hand side, you can see the outline of a plant root. These clumps that we would see here would be what we would call aggregates. And I'll get to that in more detail here in this slide. So when we get a soil test from the lab, we might get soil texture. And I would call that the primary description of soil structure. And we often think of, we use a soil triangle you can see in the bottom middle, where we can find out the fractions of sand, silt, and clay. But when we start to think about the overall structure of soil, I would say we have another tier, which is kind of what I would call secondary structure or aggregates. And this is where soil biology starts to interact with the mineral particles. And I'll just kind of walk through the steps in terms of what comprises an aggregate. On the left, we have the inorganic mineral particles, sand, silt, clay kind of things. And then we have, the glue. There's polysaccharides, so kind of like sugars that are often accompany bacteria, and that kind of helps glue these particles together. And then we have fungal hyphae. Hyphae are kind of like threads, and they can kind of knit this together. And then on top of that, we have plant roots, and then the fine roots integrating. So that combination, so I like to think of aggregates really, and actually they're kind of my favorite indicator for soil health, because they're representing not only soil structure, but also soil biology. It takes a complex food web or active food web in here to be part of the builders of that. And then these are also housing those organisms. And my macroaggregates are super important for like water holding capacity and infiltration rate into the soil. Those are just a couple properties. We also can classify aggregates in a couple different kind of general size categories. We have microaggregates that are defined here, or macroaggregates that are larger. For healthy soil, we really want a mixture of these and a diversity of ranges of sizes, and really pushing for larger ones because they're going to tell us we've got a nice balance with the biology.
I've been hearing a lot about biochar. Do you what do you know about biochar? Is it like the miracle or not so much?
Well, so that fits in with maybe this discussion because that adds structure. It also adds cation exchange capacity, CEC sites where you can, attach nutrients. It doesn't really, biology isn't really living inside there unless you're coating them, with a film of biota. So, but they are known to, help with building the soil structure. They don't do much for the biology.
It's kind of just surface area that you'd have to charge.
Yeah, absolutely. That's exactly what it is. You're just adding surface area.
See.
But it's interesting that you brought that up too, because I used to just think biochar was biochar, where in fact there's a lot of different kinds of biochar. It depends on what you make it from. And we have an assistant professor at University of Vermont who is actually investigating that and how. So if you make it from tomato waste or hemp waste, or different waste have different properties, and also the temperature you make it at can really affect the properties, the chemical and physical properties. So more than half, you won't hardly believe this, more than half of the biodiversity on Earth is found in soil. So it's many, many species, but also there's tremendous abundance. Just a few quotes that are out there is, soil is considered the most biologically diverse, as I mentioned. 1 gram, which is about the size of a green pea, contains up to 1 billion bacteria, tens of thousands of taxa, 200 meters of fungal hyphae, etc. That kind of blows your mind. Or you can think of 1 teaspoon, and a teaspoon is actually several grams. It's much more than a gram. This contains more living organisms than there are people in the world. So billions of organisms. So that's kind of mind-blowing. And that's also a reason why we should protect our soils. And when we start thinking of our soils as living, then I think that gives incentive to protect, because we want to protect living things and take care of them. So here's another way to look at biodiversity. It's kind of like this pyramid. This is a diagram you might see in an ecology textbook where they show you the different trophic groups or different groups. This diagram really focuses on kind of numbers and size of these organisms. And the largest ones are at the top, the smallest ones at the bottom. Usually the smallest are the most abundant. And so we see a hierarchy that, here is bacteria, looks like 10 trillion. And this would be, this is per area. I believe it's like 1 meter, I think. So it'd be like a yard. And it's about 6 to 8 inches deep. And protozoa, which are microscopic animals. Now, I note that there's one group here that's kind of missing, that they aren't mentioning, that would be a little bigger than bacteria. Can any of you think of what that is? I won't pause too long, but fungi.
No, I'm trying to think.
Oh, is it fungi?
Yeah, fungi would be the one I would expect to be in here, because microbes, and the word microbes gets thrown around, and it's important to clarify, as microbes mean, I think the literal translation is it's bacteria and fungi, anything that's microscopic in size, where some people use it to mean only bacteria. But I think, so it's important for people to clarify how they're using that term. When I see microbes, I think it includes bacteria and fungi. So I think they're there, and then protozoa are they are the most numerous. One of these fauna. Some people argue nematodes are the most numerous. Mesofauna, they say protozoa or microfauna. It's just differences in their sizes. We have mites, springtails, and all these different invertebrates as we go up. But it's not as simple as just saying who's there. We also want to know what they're doing. And that's kind of where the state of the art is of soil biology at this time in history, is that people are trying to conduct studies to figure out exactly what they do. And it's taken a lot of molecular biology and genetics tools to be able to do that. So I like to use the analogy of a symphony to explain how soil communities perform. A lot of times when we talk about plant pathogens or biocontrol agents that are on the market, we're thinking about single species of pathogen or a single species of a biocontrol. But we're learning more and more and realizing that actually soils work best when you were thinking about multiple species together and, that are interacting with each other. And so the way this analogy works is that basically we have all those musicians. We can pretend like those are all the species. Oh, I should point. You got all the musicians, but the thing is you have all those species, but most of them are inactive most of the time. There's only a few that are playing or active at any one time, space or time. And then the conductor is kind of the clues or cues in the environment that actually then wake up different groups at different times. So, you've got the violins playing, and then maybe you're going to bring in the trumpets later, and maybe the trombones at a different time. You wouldn't want them all playing at the same time. That would be a little chaotic. So, and then the instruments themselves, when they're playing together, is kind of like multiple species playing the same function. And this happens a lot. And the reason we want that is because they're playing the same function, but maybe at a different time, maybe a different season, or a different geography, a different ecosystem, whether it's a forest or an agricultural soil. And plant productivity is usually greatest when we have the most complex food webs. And here's an illustration of that. This is actually on a grassland system, a forage system, but they manipulated the complexity of these different food webs. And on the right two columns these are milligrams or a weight amount of carbon that we see as they would weigh the shoot or the root, and that's a measure of productivity. We can see, so they have plants without any soil organisms, or plants with bacteria in the soil, or plants with bacteria and the predators of bacteria, which would be, in this case, bacterial feeding nematodes or bacterivores. Or you can look at another component of the food chain with fungi, their predators, or if you put them all in there. And one thing we can see is the largest size of a shoot here, 13.5, is when we have the most complex food web. So we're seeing the plants are, and this suggests that there's interactions between these organisms in the soil and their interplay with the plant roots. There's a lot of, in these organisms, you got to think they don't need eyeballs like us to see who's there. It's all with chemistry, how they're communicating with each other. Sometimes my slides are changing. Here's a visual of food web complexity and how it relates to different ecosystems or different ecological succession sites. So our goal is really to get, the more complex as possible in healthy soil, as you see over on the right. Although we're not going to see that in a management system like agriculture necessarily. But so we might envision an annual cropping system to be here, like on the left. And this one would be similar to what we just saw in terms of having both bacteria and fungi and organisms that can feed on those bacteria and fungi. So that'd be a relatively healthy annual cropping system. But if we then would allow early succession to occur, like maybe we're going into no-till systems or perennial agriculture, we start to foster more components of the food web in terms of looking at omnivores and predators that might be joining in here in more complexity. And then that continues with, in a late succession system, an example of that might be, say, a native prairie. Now, one thing about soil health and the metrics of soil health is, it really depends on having a good sample. And you might have heard of the old adage, garbage in, garbage out. Well, that's so true. So one of the realities we have to deal with is that the biological communities in soil are very heterogeneous spatially, and that affects how we want to collect our samples. So on the right-hand side, we can see 3 layers of complexity. Maybe on the landscape scale, we're seeing, gradients or patches of different soil textures, carbon, etc. And we can, notice that if we look at soil maps that the NRCS produces, for example. But if we zoom in on a little component of that where it looks fairly homogeneous, it's still heterogeneous within that, and that would be at the field scale. And there's variation across the field. And if we drill down that, more at the plant level, we even see additional heterogeneity. And it's even said that if you took two soil cores side by side, you would still get slightly different results. So there's tremendous heterogeneity. So how do we deal with that? So when we're trying to sample these aggregated, patchy systems, it's really important to use what we would call a systematic design. And systematic means it's a very consistent, and I usually think consistent spatial distance between our samples or our cores, so that this is a way of reducing bias. So when I'm looking at biology, it's usually you want to have I use a random starting point, so that I'm not biased as to where I go, and maybe take my core. And depending on the size of my area of sample, I might walk 10 paces, take another one, walk another 10 paces, take another one. The idea is not to sit and think about, does this look nice or does this look bad? You're trying to be representative across that space. Unless, of course, you were worried about a particular disease or something, then you would have that would be a different kind of sampling pattern. An alternative sampling pattern is what we call stratified random. And that would be, you could take your field and develop, it could be artificial strata, like say I'm going to divide it into quadrants or maybe into 8 portions, and then take a sample at random within each of those portions. And those get mixed together as a composite sample. The composite is because we're trying to be representative across that whole area, and we need enough soil that we can extract what we need to look at. Sampling depth is also an issue. We have to think, what is our target? If we're interested in the plant roots, we want to go as deep as roots. And, the standard is usually like 6 to 8 inches deep. That came about historically because that's, kind of, the plow layer. Although now people are starting to think maybe we should be thinking about, maybe slightly deeper, but or if we're just after the litter layer, we would take more just the surface of the soil. And in fact, the organisms occupying the litter layer can be very different from in the soil around the plant roots. And for biology, we have to think about storing the samples in favorable conditions. At the very least, it's nice to have some kind of container, like a thermos or, ice chest, just to have an insulated container that you can put these samples in. If I'm looking at bacteria and fungi, I'll put in some ice. But if I'm looking at nematodes or microarthropods, I just skip the ice because sometimes ice can be damaging them. But the idea with insulated is you want to keep the temperature similar to what it is out in the field and not let it get to the extremes in temperature. We want our samples in plastic bags so they'll stay at the field moisture, but particularly extreme hot is damaging. So avoid putting them on the dashboard of your pickup or, out in the sun in the field. It's better to find some shade where you can keep this insulated container. So, because if we had extreme conditions, that could really affect the outcome or what we learn. Now, one thing I have found too is everybody kind of gets aghast at the cost of biological measurements. And I'll have to say part of the reason of the cost is because there's so much labor involved in running these analyses. Maybe we can reduce that in the future as we keep refining our methods. But, we know labor is often the highest cost in many, many efforts. So, it's not that, someone's trying to make a lot of money. They're just trying to pay for the people actually conducting the analyses. So we can either extract, okay, I'll back up. These organisms are in soil, but you can't just put soil under a microscope and see it. We have to get the organisms out of the soil before we can start to identify and count them. Traditionally, we would use culture plates, dilution plates. Now, bacteria are so numerous, they're in like billions per gram of soil. And so we couldn't put billions on a petri dish and expect to be able to do any counts. It'd just be one big mass. So we have to dilute it down, and we end up diluting it tenfold for multiple times until it is about to 1 billionth. And then we can plate that on a petri dish, and then we can try to identify the colonies. The problem is only about 10% of the organisms in soil actually can grow on a petri dish. So we've missed a lot of organisms there, and the current molecular tools are allowing us to find them, find organisms that we were missing before. And that's giving us a completely different picture of who's there and how many. Another approach is using DNA analysis, and there we can take a small portion of soil and run it through a series of solutions to extract the DNA. And other methods for like microinvertebrates are illustrated more on the right-hand side. We might make a soil slurry and run through a series of sieves. That'd be for nematodes. Microarthropods, we can use a method called a Tolgren funnel or Berlese. And in this case, we have a funnel with a heat lamp on top, and then there's a screen on top of that funnel and a container at the bottom to collect them. So microarthropods don't like heat and they don't like dry. So you're exposing them to both heat and dry, and then they try to run away. They fall through that mesh from the funnel down into the container. Sometimes we want to run bioassays, especially if we're trying to screen for disease like soil-borne pathogens. The microarthropods, still we use a lot of, and nematodes, we still use a lot of microscopy. For identification of bacteria and fungi, we have to use more on the genetics and biochemistry as tools. Just wanted to introduce you to a few of the players in soil. Some of these are probably familiar and others kind of different. So I'm just going to have two columns here. One is kind of bacteria on the left and fungi on the right. Bacteria are really kind of boring to look at as far as their morphology or their physical appearance. They're either cones or, they're spheres or, like rods. So that's kind of boring, but where they really are diverse is on the biochemistry. So that's why, we have to use molecular methods, which is applied biochemistry to be able to differentiate these. We can do some on petri dishes, but a lot of them not. So we know, and the thing is we have to, I've just identified these two genus, but there's a lot of diversity within genera. Like Pseudomonas is one that's common, but you have species that can be pathogens, you have species that might be biocontrol, you have species that can break down organic matter. So you really need to know which species you have in the conditions. Another one that fits in the same phylum is Rhizobium, and this is the bacteria. This genus has species that are involved in nitrogen fixation. We have Firmicutes is another phylum of bacteria. And again, there's, even with Bacillus as a genus, there's Bacillus that cause diseases like anthrax or botulism. That's why we have to have high heat when we're canning them to kill that, because these organisms have really thick survival spores called an endospore, and that allows these bacteria to survive a long time. But on the other hand, we have these two that are considered biological control agents. We've got BT, the Bacillus thuringiensis, or Bacillus subtilis. Another bacterium used to be classified as fungi are the actinobacteria. These are very slow growing. They tend to grow more in alkaline soils. But they have a variety of functions they can play. They can be saprophytes, parasites, sources of antibiotics, and they're also the organisms that kind of give soil the earthy smell. On the right are the fungi. We have yeasts, which are named kind of this category because they don't have any reproductive structure. Most taxonomic keys rely on having a reproductive structure. Yeasts are used in fermentation, for example, Saccharomyces, for brewing beer, baking bread, etc. Then I'm not going to talk too much about the Chytridomycota, but these are organisms that are fungi that can be found in waterways. But Zygomycota is an organism these groups are involved in decomposition. Rhizopus is one that you might see on your bread if it's starting to look a little moldy, kind of thought of as the bread mold. There's also a different species of Rhizopus that's used in making tempeh, which is, it's a process where you're doing mild decomposition and, the microbes are imparting the flavor. Then there's a phylum called Ascomycota, and in there we have Alternaria. There are some Alternaria that might be decomposers. There's some Alternaria that are pathogens. Penicillium, of course, very famous for its production of antibiotics. But generally in soil, it would be more of a, it would be more of a decomposer. But the fact that many microbes produce antibiotics, that's just a means of defending themselves. They have to have a way of chemically communicating with other organisms and be able to figure out who's a friend or foe. Trichoderma is also an Ascomycota. This has been used for many years and sold as a biological control agent, and it's a heavy sporulator. Then mushrooms, we always know those are fungi, but that's really just a single phylum called Basidiomycota, and that fits in there. So, many times I said that, there can be multiple functions within one of these taxonomic groups, but that also just implies we need to identify them to the finest level of taxonomy as possible. If we just say fungi, it can be a lot of different fungi. We need to know more about who is there and what functions are they playing. Then we have protists. There's kind of four major categories of protists. We, illustrated on here, we've got those that are called the phagotrophs, and these are feeding on other microfauna and interacting. There's the other class of symbiotic ones. They can be involved in diseases, or they might be mutualists. We see the pathogen Phytophthora fits in this category. That'd be an example of a pathogen. Where we have those that their primary function is degrading organic matter. Or we also have another group that can photosynthesize and fix carbon, and this would include algae as well as diatoms would fit in this category. Now, when we think in the soil, in general, in terms of interactions, there's really an interdependency between plants and the soil food web in the soil. And, the organisms, there's a heavy concentration of them anywhere around the root, and we call this region the rhizosphere. And it ends up as plant roots grow, they're kind of leaky, and they exude amino acids and sugars. This provides a lubricant for the roots to, penetrate into the soil. So that's why the plant's doing it, but these end up also being food for the microorganisms around there. So that's why they're concentrated there. And if we were to divide the microbial community into kind of three big classes, three big classes is we have those that really have no, or kind of a neutral relationship with plants. We have those that are beneficial, and the interaction with plants will assist them in taking up nutrients as well as defending the plants. Like some microbes might send a chemical signal to the plant where they put out defense mechanisms, or like this one is called an induced systemic resistance, where it spreads defense throughout the plant, indicating there might be an attack. And then we have the organisms that are pathogenic, and they may be toxic or causing a disease of plants. And then there's interactions between these. And back to the complex food web, this interaction is really important. If we only had pathogens, they could take over, but it's really this balance of other beneficials and these neutral players that can kind of help keep the pathogens in check. So, you know, and I often think of the gut microbiome. We've learned, you know, it's really important to have a diversity of organisms there so that the pathogens will kind of, you know, be controlled by having that balance instead of getting, you know, the imbalance is when we have disease.
Now, when you're thinking about,
go ahead. Yeah.
when you think about nitrogen fixation, you know, especially the legumes, are those microbes dependent on those root exudates, or does that occur to some extent just on their own with sugars that happened to be in the soil?
Oh, that's a really good question. And if we want to get into it more in the Q&A, I have a picture that kind of shows it. So there's a very specific relationship, and it's more based on chemicals that are exerted by the plant and the root, and they find each other. There's a compatibility. There's certain strains that'll match with certain varieties, plant varieties. I mean, it can be very specific, and back and forth on the communication, then they know that that's compatible. So it's not like, and then once it's compatible, then there's these signals that go in terms of the bacteria actually able to get into the plant root. The root responds by curling around it, and then it can actually get inside the plant root. And then there's communication where you get this proliferation of cells. So it's a very specific relationship. It's not just, you know, broad like this, but what's kind of cool is in a way it develops this factory that's converting nitrogen gas from the air, you know, into ammonium, which is a product of nitrogen fixation. That's a process that happens to happen in anaerobic conditions. No oxygen. Well, there's oxygen in most soils. We want that. But so these little factories actually have a structure around it that maintains an anaerobic environment inside for these bacteria to operate. So it's kind of cool.
That's pretty cool.
Yeah. Okay. I know I've been talking a while, but I wanted to just I could continue on indicators themselves if you have questions. I have slides on that, but I did want to tell you about a new service that we've established just 1 year ago at University of Vermont. And, you know, we're starting with a focus on Vermont clients, but we intend to, you know, pretty much serve New England. And we're calling it SHREC as a acronym standing for Soil Health Research and Extension Center. And our goal is really, a focus is really trying to serve farmers. During the pandemic in 2020, we got a group of us across the campus at University of Vermont that kind of did an inventory of what the options are for soil health tests and how farmers responded to the different data they were receiving. And what we learned is they felt like there was a lot of data they were receiving that was hard to interpret or wasn't necessarily reflecting how they saw they were adjusting their management practices. And they also wanted more biological measurement. So we set this up as the mission of SHREC to really try to focus on introducing more biological measures and also those that we know respond well to different management practices so that we can improve our calibration and interpretation and be more regionally specific. With that, I wanted to just share with you just a brief 3-minute video that kind of introduces this center. So.
I'm not getting the audio.
Yeah, we're not getting any audio.
Oh, okay.
Possibly because it's through your headphones.
Oh, yeah. Let me try. Off. See.
A laboratory for
Does that help?
50 plus years.
Yep.
Focusing on soil testing.
Let me back it up and we'll just start.
Farmers are asking. Sustainability really starts with soil. Soils really play a lot of really important ecosystem functions. if we take care of them. But if you can manage your soil well, that's like wealth. University of Vermont has had a laboratory for 50 plus years focusing on soil testing. Two big things the farmers are asking: tests and results that reflect their management practices. And secondly, to include more measurements that reflected the biology of soil. So those, I think, are two pillars that really develop the mission or the vision behind SHREC.
In the past, farmers in Vermont could send their soil samples out of state for testing, but it wasn't informed by land grant knowledge and calibrated just for Vermont soils. SHREC is an opportunity for Vermont farmers, Vermont extensionists, Vermont researchers to get Vermont calibrated information for Vermont soils, Vermont farming systems, and the Vermont climate.
Working with farmers, they live and breathe and, work it. And they've given me a lot of good ideas that led into important research projects for myself. And I always really valued that because I felt like then I knew what I was doing was really going to, have an impact. So the way the lab works is the farmers will receive instructions and guidelines about how to collect the sample and then mail it or drop it off at the lab. The lab is analyzing the soils and then preparing a report that includes the results as well as some background materials about what that means and management practices that can be implemented to change it.
In SHREC, we offer information on not just how much carbon is within their soil and organic matters within their soil, but what types of carbon and what types of organic matter are in the soil, and that matters. We're also providing some information about aggregate stability. So how well does a soil hold up to the impacts of heavy extreme storms, which we're seeing more of with climate change. Help farmers understand what practices they can do to help minimize erosion and soil loss from their land. We're also providing some information about ACE proteins, newer tests that helps farmers understand what the total amount of nitrogen is available within their soils.
Another component, obviously, is education. We want people to understand the value and, what these measurements are and what they mean, and we need feedback. So we're offering workshops, focus groups, field demonstrations where farmers are going to be full participants in that. We like to have that feedback. That it's a win-win situation.
We're not just producing test results for researchers. Research is very important and it'll be part of SHREC's mission, but ultimately we want farmers to be able to access the tests we're providing and to use that information and really put it to use in their farming on a day-to-day basis. Having healthy soils is really critical for a thriving food system. I think the future direction of soil health testing is that we're going to need very local information, state-based information, and I think we're in a very good position to do that for our farmers here in Vermont based upon our farming systems and our climate.
So that is, you can see the the web address for that lab. And I could go on about the tests that we run if you'd like to, but I don't want to. I want to just kind of finish here with a recap of some of the main points. So one is to keep in mind that soil is alive and that food webs are complex and that complex food webs are healthier than simple food webs and they're more productive. And if I got into the indicators, I could convince you that it's really most cost effective to think about picking those that are complementary, telling us different things rather than multiple ones that tell us the same thing. The interpretation of microbes will depend on the trophic groups above it. So the organisms that are feeding on microbes or predators will influence what's happening at the microbial level. And some of the initial offerings that we have in the lab are aggregate stability, and active carbon, an organic form of protein, respiration. We're going to do fungal to bacterial ratios. I think with that, I'm just going to end and turn it over into Q&A.
That's good. Paul, did you have a
Perfect.
question?
I do, but they're kind of just like my personal office hours, so you might as well end the
Okay.
recording.
I was going to ask,
Yeah.
so like a lot of this work has to do with like the food system and so like farm, like that kind of scale. For myself,
Yeah.
who is not a farmer or grain grower, like, but I am a gardener, what would be my like any particular key takeaways that would be um specific to somebody who is like just has a home garden?
Yeah. Well, I mean, home garden, I mean, of course, the first thing we always think about is make sure we have enough nutrients
Hmm.
there. I think, and that's been the tradition in history, and that's certainly important. But we kind of want to know, we want to know what the tests tell us kind of where we're at, in terms of do we have enough or do we have too much? We don't want too much because that's a problem because you can have runoff. On the other hand, I think, gardens, I think about really trying to get that organic matter into that soil is really key. And I think that's something even key on larger scales too, that with our management practices that we've had globally, too, with tillage and all this stuff, we've lost a lot of that, but provides such a service um, in terms of providing the microbes. It gives them the habitat. It helps with water holding capacity, for drought. And it can be a mulch. I know I use a lot of mulching on my own garden. And then that really it helps make the garden more drought resistant. You don't have to, and it also helps keep the weed pressure down. Um, and providing that complex. So I think the same principles can really work at a lot of different scales.
Hmm.
You might not, um, you may not want to pay thousands of dollars to get every biological test. But one simple way you could do is look at your soil and how crumbly the structure is. And if it's crumbly, that the crumbles part is the aggregates, and so that's going to tell you have um, you've got life in there. You've got the microbes, you got, that's building that. And that's a good thing. And you don't want it like, so it looks like it's pulverized or something. That would be bad. Not so good holding it in place. So those would be a few things with the gardens. What was I? Oh, one thing that you hear a lot about is earthworms. And I just wanted to say here in New England, that's a little more complicated story because our areas underwent glaciation, and when the glaciers receded they basically wiped out most of the native earthworms. So most of the earthworms we see in New England are actually invasive and were introduced you know um, from immigrants, people fishing and so forth, and so some of those can be detrimental in terms of really, like in forests, they're finding, like in maple forests, for example, those really go through the organic matter and are taking it down below the surface. And that's really changing the structure of the, say, the forest floor. So it's drier now and warmer. So they're actually finding maple seedlings are having a harder time um, getting established and maintaining our maple forests if we have these invasive earthworms in these systems. So it kind of turned things upside down because, generally we think of things like that as really being the engineer and changing the, water infiltration patterns. But it's kind of a double-edged sword, I guess.
That's
Breaking down the
interesting.
leaf litter too fast.
Yeah, what?
They bring down the leaf litter too fast.
Yes, yeah. So if you find side by side the forest, one with and without, the ones without will have a nice thick leaf litter, and the other one you can see a much thicker, organic layer under the surface. But it just changes, you're kind of losing that mulch on the surface, which is a factor, and it's just not conducive to the establishment of the tree seedlings. It's going to favor a different system that can tolerate the heat and the dry. So
Remind me, I was in the rainforest in Costa Rica and in Ecuador, and And it's like, yeah, soil's horrible because like it's just
Yeah.
everything just disappears immediately when it hits the ground.
Yeah, yeah, there you go. It's kind of like that phenomenon. There it's for a little different reasons. It's
Yeah.
warm, it's very moist, so that's ideal for
Yeah.
decomposition, right? Thinking of that, I did a project down in Costa Rica in the rainforest. We were looking at nematode community and how many different species we'd find. And because the scientific literature said we actually had more diversity on the North Pole and in the Arctic than we did in the tropics, which is opposite of what we would expect.
Yeah.
So we were like, okay, we got to see if this is really true. And I thought it's a sampling problem, isn't it? Because we always sample soil. And as you know, the soils get really thin there, right? But if we sampled above ground in the epiphytes and all the, mosses and it could be orchids or ferns or whatever they're growing on, these all there were nematodes in all of those. And if we added all that up, we had equal or greater
Oh, wow.
diversity. So it was like, we got to be looking in the right place. Where are they? And
Start
if we
studying the
only,
tree canopies.
yeah, exactly. We wanted to go higher in the canopy. About all we could do is about, 9, 10 feet up, which is
Right.
nothing compared to how tall those trees are. We just need to be able to climb up into the canopy. That was what we wanted to do in the long term, but we didn't get the funding to do that. But they have found in other organisms, I know people that have studied mites and columbella up in those canopies, and that adds an incredible diversity. There's a lot of niche differentiation as you go, up from ground to the canopy.
That is cool. I just planted this field. It's my first year growing on it. It's right on like I want to say it's an old floodplain near the Kennebec River. So the soil's like almost all silt. It's like the siltiest soil I've ever
Mm.
seen. Every time it rains, water table comes up immediately, turns into quicksand, and then it immediately drains when it stops raining. So it feels really nice to work with, but I feel like there's no structure there.
That's
Um,
that's true. Yeah.
and I'm planning to grow there for, the future. And I'm just like, what do I do? This is year one. Like, how do I get this structure built into the soil with it and not deplete it? Because I'm growing grains.
Well, I guess, one thing to think about in terms of establishing it is I think of getting if you can get some plants on it and keeping plants on it continuously, that's going to bring a lot more, life and organic matter into the system. Perennials even more so, if you could get some uh whether it's, some kind of a shrub or fruit bearing or grasses, because all of those roots are going to keep adding nutrients and organic matter. Plus, the roots are going to help hold it in place and start. So that's one thought. Um, I'm just thinking back to my own, because you're trying to get a better balance on there. Organic matter is going to be one, would be the main thing you could do. I mean, you don't really want to haul in a lot of clay, but you're trying to get some kind of mixture. You need more of a
Right.
mixture of sand and clay and silt. If you could just get a silty sand, it would be better than just
Yeah.
silt.
Yeah.
But then,
It's
you got water flow. Sands don't hold water very well.
no. I mean, and it's weird because like last year, I had a field up near Bangor, almost all clay. Like I could spin a pot with that
Yeah.
soil. And it was just such a nightmare. I stopped growing on it. And now I got this silt and it like feels like a miracle that I can actually like, you know,
Yeah.
it water flows through it.
So you just need something
Um,
there. I have worked with an organic farmer here in Vermont who he started his farm on pretty heavy clay. And I went on there and said, "This is the greatest tilth I've ever seen." And if you stepped on it, your foot would sink a few inches. He has added so much organic matter, it changed the soil. So, but he's been doing, took him 20 years to get to that
yeah,
point, and continuous. But since, he also protects that soil, he's like, I don't want you walking on my field.
right, yeah,
So we were always very careful.
yeah, yeah. I just cover cropped this new plot, the silty one, with I meant to do buckwheat, but Fed messed
Yeah.
up my order back, and I got a bunch of
Oh
oats, so I'm like,
no.
oats will do. Threw a bunch of oats
Yeah.
and sun hemp
Just
on
get
there.
something growing in there. And, there's a lot to be said for trying to keep some kind of continual cover where I mentioned perennials, but if the cover crops, that kind of thing too will help in terms of holding it and also trying to get more of that infiltration. I don't have much direct experience just with strictly silt, just more.
Yeah,
Uh,
there's other stuff in there, I'm sure, but it is very silty.
yeah, and
It's got that
brings up.
quicksand element to it. It's
Yeah, you need a little bit more variation of
yeah,
materials in there.
right.
But I guess the one thing I think you could control is thinking about organic matter. But you can't just dump like lots of loose compost on there. You need something to help hold it in place, in case you
Right.
get the rainfall or um, I know we've been struggling with flooding here in the last few years too, which could be a real
Yeah.
nightmare in those systems. Are you near surface water like creeks,
It's
rivers?
right along the Kennebec River, so
Oh, that's
um
right. Yeah.
I imagine it
Which,
used to be the riverbed and now it's the river's moved out of that area, but um yeah, within eyesight of the river.
yeah, that's why it's so silty then.
Yeah.
Yeah.
And very high sulfur for some reason, which I haven't figured out why.
Oh,
Like
interesting.
very, very high sulfur.
Yeah, you don't know if it's a natural source or if it's from something human made, but
Yeah, I was wondering maybe if it was pesticide runoff from the logging industry up in like way up north in Maine. Um, but I don't
if
know
they
if,
would use,
are there
yeah.
any sulfur-based pesticides or anything they're using? Because that would be like that's the only kind of like or paper mills. There's a lot of paper mills
Okay,
on the
that could
Kennebec.
be it. That sounds
That might
almost,
be
I mean,
it.
my gut feeling is that might be more of it.
Yeah,
But how's your pH? I would think paper mills, I think to
pretty
make it
acidic.
acidic.
It's like 5.8.
Yeah.
Um, and I just limed it, so hopefully I got it above 6.
Yeah.
And,
Yeah. Liming,
yeah,
that'll be important too to get better productivity because yeah, the cereals aren't going to want to live in that acidic soil. You could
right.
grow blueberries fine there.
Yeah. Yeah, blueberries are great here. Yeah,
Yeah.
one plot there's dug up two plots. One of them I just cover cropped immediately, kind of prepping it for next year.
Yeah.
It was fallow before, and it was mostly for the weeds. Like I just want to smother the weeds for next
Yeah.
year, and get some organic matter. And then the other one I'm gonna grow. So I limed that one. It was started 5.8. I'm just like, oh, if I get it to 6 point something, that'll be good for.
Yeah. 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 That'll get you, yeah, close. And your idea of the buckwheat's a good one. I've seen that. I've heard many cases of that being a very good weed suppressor.
Yeah, yeah, nice broadleaf shade. Though
Yeah.
now we got oats, so
I
we're gonna see
don't
how
know
the
what
oats do.
I don't know what the market is for buckwheat.
I'm just gonna till it under.
Okay. Well, that's
So
the other thing is
actually, I
just
could tell you the
green
market is for
manures.
buckwheat. I work at the mill, so we sell buckwheat. It's booming.
Yeah, yeah, and or green manures in terms of just yeah, like is how you're treating it. It sounds like
Yeah.
yeah,
I wish I had a roller crimper though, so I didn't have to.
yeah. Wouldn't that be nice? And
Yeah.
then you could roughen up the surface. Then you could keep more materials there from and
Yeah.
not running into the river.
Right.
And that I would think would help build the soil too.
Yeah, and it'll keep a good ground cover. You don't have to till it again. And then
Yeah,
if it's laying them all down in the straight like same direction, you could just plant right into that. Whereas if I just like mowed it down, it's gonna be cross-hatched and that'll be like a thick
yeah. I think yeah, you
mat.
yeah, because you want your seed to be able to come up through that.
Right.
Yeah,
So I might have to till it under there because I don't have a roller crimper.
yeah. I don't know too many people that do. Isn't it pretty? It's pretty new,
Uh,
that concept.
UMaine's got one,
Okay.
the UMaine
And I know
farm.
Cornell
But
has one.
yeah, it's probably the price of a house, I bet.
Yeah, it's one of those things you probably wanted in a sharing setup. So if you had
Yeah,
one, several people could use it.
yeah,
Yeah, but it seems like a promising way to keep, you keep your surface roughness and also you get a good source of organic matter
yeah,
there. I feel like one of the big issues, yeah, in our agricultural systems is we've really limited, we don't have enough organic matter. And I've worked in it like a disease suppressive system in the Midwest, like in Minnesota, and they actually found they had a very high percent of organic matter, but they found they got some natural disease suppression that established over time in that setup. They did no-till soybeans and this high organic matter, and they actually had natural suppression to soybean cyst nematode.
Oh, really?
Yeah, which is a
Oh,
tough one, and it keeps
yeah.
spreading more northeast. And so we tried to figure out if how soon as we had treatments of like crop rotation or we tilled it, or used some kind of pesticide, the suppression all went away.
Really? So crop rotation made the suppression go away.
Isn't that
Yeah, that
weird?
is
It's
weird.
opposite of what we
Yeah.
learned, right?
It's opposite of what
Yeah,
we tell everyone to do.
that's right. And and me being trained as a plant pathologist, that was like number one. You
Yeah.
got to rotate it away from your host plant. I think what happened is at least my hypothesis was that by keeping it no-till, high organic matter, their disturbance level was relatively low. All they had was their planter, and it probably had an adaptation. It kind of adapted and came to a nice equilibrium.
Right.
And then any change you made, threw off that equilibrium.
Huh.
That's my guess.
Yeah. I mean, it would make sense.
But it was quite an unusual system. But it convinced me that higher organic matter could really be helpful, not only in like water infiltration and holding capacity, but also providing more complex, being the base of a more complex food web, and diversity of organisms that could help keep pathogens in check.
Mhm.
Hmm. Well, I do not know where Seth is. Um, I actually
Yeah, I'd probably
just
just text him the link in case he was expecting a separate
okay. I
one.
did actually send him an email, but if you text
Okay.
him, just see if he wants to hop on. But, actually, am I
Uh,
into the recording here?
yep.
Sure.
We could continue talking, but so let me see how to do that.